Think about walking through a garden. You see all these different plants, some with broad leaves, some with needles, some with sprawling roots, others with tall, slender stems. They look so different on the outside, don't
Yeah,
they?
absolutely.
But it's like looking at different models of a car. The exterior styling changes, but underneath, there's a fundamental engine, a chassis, a way of converting energy into motion.
Or different dog breeds, right? A tiny terrier to a giant mastiff, same basic blueprint, just executed very differently.
Exactly. And that's what we're digging into today for you, someone who wants to get to the heart of things quickly, but thoroughly.
Exactly. Today's deep dive takes us into that hidden world inside flowering plants. We've got some really fascinating material here that systematically peels back the layers of their internal structure.
Okay.
Our mission really is to uncover the core building blocks, the cells and tissues, and understand how they're organized to make these vital organisms work, how they thrive.
Okay, so if you really wanted to understand how a plant works, just looking at the leaves and flowers wouldn't cut it.
Not even close.
It's like trying to understand how a building stands tall just by looking at the facade. You'd need the blueprints, wouldn't you?
Yeah, you absolutely would.
You'd need to see how the foundation is laid, how the load-bearing walls are constructed, how the plumbing and electrical systems are integrated.
That's the level of detail we're aiming for with flowering plants today.
Got it.
Precisely. This internal organization operates on several interconnected levels. It starts with the cell, the fundamental unit of life. Think of these as our individual Lego bricks, maybe.
Okay, bricks. I like that.
Then, groups of similar cells working together form tissues, like using those bricks to build a specific wall or, a support structure.
Right.
And finally, different tissues combine to create organs, such as leaves specialized for capturing sunlight or roots designed for absorbing water and nutrients.
So cells make tissues, tissues make organs.
Exactly. Each with a specific role to play in the plant's survival.
Right, like those brick walls and support beams coming together to create a room with a specific function, and then multiple rooms forming an entire building.
Perfect analogy.
And it's not a one-design-fits-all situation, is it?
Yeah.
Even within the same plant, the internal structure of a leaf is completely different from a root because, well, they have such different jobs.
Absolutely. And what's truly fascinating, I think, is that even within the vast group of flowering plants, the angiosperms, we find significant internal structural differences.
Oh, yeah.
Consider the two major groups, monocots, like grasses and corn, and dicots, things like beans and roses.
Okay.
They're both flowering plants, sure, but their internal organization is as different as the blueprints for, say, a simple ranch house versus a complex multi-story building. Fundamentally different layouts.
Wow. So it's not just different rooms within a similar structure, but almost like comparing fundamentally different architectural styles.
You got it.
And I recall reading that these internal structures can even adapt to the environment, like a desert plant being internally engineered quite differently from one in a rainforest.
Exactly right. Environmental pressures drive these adaptations. A desert plant, for instance, might have specialized water storage tissues in its stem, a feature you wouldn't typically see or need in a plant living somewhere consistently wet.
Makes sense.
It all comes down to structure-enabling function, especially when facing environmental challenges.
Okay, we've got the levels. Cells, tissues, organs. Now, let's zoom in on tissues. What exactly defines a tissue in a plant beyond just being a collection of cells?
Right. A tissue is more than just a random jumble of cells. It's a group of cells that share a common origin, meaning they likely came from the same initial cells through division, and crucially, they work together to perform a specific function or set of functions.
Okay, common origin, common function.
I'll think of it like a specialized team in a factory, maybe, say the painting team. They probably had similar training, that's the common origin, and their job is specifically to paint the product, the common function.
Got it. So there's a shared lineage and a coordinated purpose. And I understand there are different categories of tissues based on whether their cells can still divide and contribute to growth.
Precisely. Plant tissues are broadly categorized into two main types. Meristematic tissues, where cells are actively dividing and responsible for generating new growth.
The growth zones.
Exactly. And permanent tissues, where the cells have typically matured, specialized for particular tasks, and generally lost the ability to divide.
So kind of like trainees versus specialized workers.
That's a great way to put it. Trainees who are still learning and developing the meristematic cells versus the seasoned craftspeople with specific skills, the permanent cells.
Meristematic tissues, the plant's growth engines. the construction zones. Where exactly are these active areas?
Plant growth is really concentrated in these specific regions called meristems, regions of intense cell division. The name itself comes from the Greek meristos, which means divided. Tells you everything.
Ah,
Think of meristems as the construction sites of the plant, constantly churning out new cells that will eventually form all its different parts.
and the apical meristems are the ones right at the very tips driving growth and length, right? Like the top
Yeah.
of the shoot and the bottom of the root.
Exactly. Apical meristems are found at the apex, the very tip of both roots and shoots the stems. Their main job is primary growth, which is the increase in length.
Growing taller, growing deeper.
Precisely. This is how roots push down into the soil to find water and nutrients, and how stems reach up towards the sunlight. Our source material even mentions the figure 6.1 showing these locations.
Right.
Imagine a skyscraper being built taller by the crew at the very top, that's the shoot apical meristem, or a tunnel being dug longer by the drill at its leading edge, that's the root apical meristem, always extending the structure.
So apical meristems handle the main up and down growth. But what about those little buds you often see in the angle between a leaf and the stem? They seem important for branching out.
Ah, good point. Those are the axillary buds. As the shoot apical meristem grows upward and makes leaves, some of those meristematic cells get kind of left behind at the nodes, in the axils that angle between the stem and the leaf stalk.
Okay, little leftover bits of growth potential.
Exactly. These leftover cells form the axillary buds, and each one of these buds has the potential to develop into a whole new branch, or maybe a flower later on.
Wow.
It's like having spare construction kits tucked away on each floor of our skyscraper, ready to be activated to build out a new wing or add a balcony.
That's a fantastic analogy. Okay, so apical for vertical, axillary for branching. Are there other types of primary meristems involved in this initial lengthening? I seem to remember something about grasses.
Yeah.
How they can regrow after being cut or grazed.
Yes, absolutely. That regrowth in grasses is largely thanks to intercalary meristems.
Intercal.
Unlike apical meristems at the tips, these are located between mature differentiated tissues, often at the base of nodes or leaf blades in grasses and similar plants.
So not at the very ends.
Right. And their crucial role is regeneration after damage, like grazing. If a cow chomps off the top of a grass blade or you mow your lawn, the intercalary meristems lower down can kick into gear and push out new leaf tissue from within the mature parts.
Like having repair crews stationed along a highway. If one section gets damaged, they can fix it without rebuilding the whole road from the start.
Exactly like that. It allows for really rapid recovery.
Okay, so apical and intercalary meristems both contribute to the plant's primary growth, its length. What about when plants, especially woody ones like trees, get thicker, wider?
Ah, that's secondary growth. That increase in girth or thickness is driven by secondary or lateral meristems.
Lateral, meaning sideways growth.
Yep. These meristems are found in the mature regions of stems and roots, particularly in dicots and gymnosperms, think trees and shrubs. They are typically cylindrical in shape.
With a layer.
Kind of. There are two main types. The vascular cambium, which produces new xylem and phloem, making the wood and inner bark.
The rings in a tree trunk?
That's largely the vascular cambium's work, yes. And the cork cambium, which produces the outer bark, the cork. These usually appear later in the plant's life than the primary meristems.
So back to the skyscraper analogy.
Once the skyscraper reaches its main height using the apical meristem, the lateral meristems are like crews that start adding layers around the existing structure, making the whole building wider and more substantial.
Primary meristems for length, secondary for width.
Oh.
Got it. Now, all this cell division in the meristems is creating floods of new cells. What happens to them? They don't just keep dividing forever, do they?
No, not at all. Once a cell is produced by a meristem, it usually undergoes a process called differentiation. It specializes.
Takes on a specific job.
Exactly. It changes its structure and function to become suited for a particular role, maybe transporting water, storing food, providing support, whatever the plant needs. And crucially, as they specialize, these cells typically lose their ability to divide further.
They settle
You could say that. These mature, specialized, and generally non-dividing cells then form the permanent tissues of the plant.
Like our construction workers, once they've built a wall, they become part of the wall, not active builders anymore.
A very good way to think about it. They transition from active creation to being part of the stable structure or system.
That makes sense. So from the active meristematic cells, we get these stable, specialized, permanent tissues. And the material mentioned that during that initial primary growth, The apical meristem gives rise to the main types of tissues. What were those again?
Right. As the apical meristem divides, the new cells differentiate into what we call primary meristems, which then give rise to the main tissue systems. There's the protoderm.
Protoderm. Sounds like fur skin.
Exactly. It develops into the dermal tissues, the outer protective layer. Then there's the ground meristem.
The filler.
Pretty much, yeah. It produces the ground tissues, the bulk of the plant body. And finally, the procambium.
Procambium. Leading to cambium, maybe? Like vascular cambium?
You got it. The procambium develops into the primary vascular tissues, the first xylem and phloem for transport. These three, protoderm, ground meristem, procambium, are the precursors laid down during primary growth.
Okay, so we've seen how the plant grows thanks to those meristems. Now, let's get into the permanent tissues, the ones doing the specific jobs day to day. The key thing here is they've generally stopped dividing, right?
Exactly. The defining feature of permanent tissues is that their cells have matured and specialized. They've kind of retired from the cell division game to focus on their assigned tasks.
Back to our specialized workers.
Right. And these permanent tissues are broadly classified based on the types of cells they contain, simple or complex.
Simple and complex. What's the basic difference?
Simple tissues are, well, simple. They are made up of only one type of cell, all working together on a similar function. Imagine a wall built entirely of standard red bricks. That's like a simple tissue.
Okay, one cell type.
Complex tissues, on the other hand, are more intricate. They consist of multiple different types of cells, all working together as a team to perform a specific, often more complicated function.
Like the electrical system in a building, wires, switches, breakers, all different parts working together.
Perfect analogy. Xylem and phloem are the classic examples of complex tissues in plants.
Okay, let's tackle the simple ones first. One cell type doing one main job. I know there are three main kinds.
Yes, the three main simple tissues are parenchyma, collenchyma, and sclerenchyma.
Parenchyma. That name pops up everywhere when you read about plant anatomy. Seems like it's the most common.
It really is. Parenchyma is incredibly widespread and versatile. It forms the bulk, the main filling, of many plant organs: stems, roots, leaves, fruits.
So the packing peanuts of the plant world.
Haha, maybe a bit more functional than packing peanuts. But yes, it fills space. The cells themselves are usually roughly spherical or cube-shaped, what we call isodiametric. They have relatively thin cell walls made of cellulose.
Thin walls.
And they often have intercellular spaces between them, little gaps that help with gas exchange and stuff like that.
And what do they actually do?
A whole range of things. If they have chloroplasts, they perform photosynthesis like in leaves. There are major sites for storing food, like starch or fats. They can secrete substances like nectar or resins. They're really the general-purpose workhorse cells.
Adaptable all-rounders.
Got it.
What about collenchyma? I associate that with support, especially in young, growing parts.
You're spot on. Collenchyma's primary role is flexible mechanical support. Think of it supporting young, growing stems, leaf stalks, petioles, and along the veins and leaves, parts that need to be strong but still able to bend and grow.
Where do you find it usually?
Often just below the epidermis, especially in dicots. It might be a continuous layer or just in strands or patches.
And what makes it supportive but flexible?
The key is its cell walls. They're unevenly thickened, especially at the corners, with extra deposits of cellulose and pectin. This gives strength, but because it's not uniformly thick and doesn't have lignin, it remains flexible.
So strong corners, but not rigid all over.
Exactly. And interestingly, collenchyma cells are alive at maturity and often contain chloroplasts, so they can chip in with photosynthesis too. Unlike parenchyma, they usually pack together tightly with few intercellular spaces.
Okay, so they're like the flexible scaffolding supporting growth without being brittle. That leaves sclerenchyma. The name sounds hard.
It means hard. Sclerenchyma cells are all about providing rigid strength and support, especially to mature parts of the plant that have stopped growing.
How do they achieve that rigidity?
Their defining feature is very thick secondary cell walls that are heavily impregnated with lignin.
Lignin, that's the stuff that makes wood hard, right?
Exactly. This makes sclerenchyma cells incredibly strong. They're often dead at maturity. It's really just the tough, empty cell wall left behind providing the structural support.
Like the reinforced concrete or steel beams in our building analogy.
Precisely. Maximum strength and rigidity. And sclerenchyma comes in two main flavors: fibers and sclerites.
Fibers and sclerites, what's the difference?
Fibers are typically long, slender cells with pointed, tapering ends. They usually occur in bundles and provide tensile strength. Think of the fibers in rope or linen fabric.
Like threads providing strength when pulled.
Exactly. Sclerides, on the other hand, are much more variable in shape. They can be short and blocky, star-shaped, all sorts. Their main feature is extremely thick, heavily lignified walls, often with obvious pits, and a very small internal cavity or lumen.
And where do we find these?
They're responsible for the hardness of things like nutshells, seed coats, and they cause that gritty texture you sometimes get in pears or guavas.
Ah, that grittiness. Okay, so fibers for tensile strength, sclerides for hardness and compression resistance. That covers the simple tissues. Now, for the complex ones, xylem and phloem, the transport systems.
Exactly. Complex tissues, multiple cell types working as a team. Xylem and phloem are the plant's long-distance transport highways.
Let's start with xylem. What's its main job?
Xylem has two key functions: conducting water and dissolved minerals absorbed by the roots upwards to the rest of the plant, and providing mechanical strength and support.
So plumbing and scaffolding, basically.
That's a great summary, and it's made up of several different cell types working together.
Right, not just simple pipes. I remember tracheids and vessels being key components.
Correct. The main water-conducting elements are tracheids and vessel elements, which form vessels.
Tracheids first. What are they like?
Tracheids are elongated cells with thick, lignified secondary walls and tapering ends. They're dead when functional. Water moves between adjacent tracheids through pits, thin areas in the walls. They're the main water conductors in gymnosperms, like pines, and also found in angiosperms. Think of them as relatively narrow, sealed pipes connected by these pits.
Okay. And vessels. How are they different?
Vessels are generally considered more efficient water conductors. They're found primarily in angiosperms, the flowering plants. They are formed from individual cells called vessel members, which are typically shorter and wider than tracheids. These vessel members are joined end to end to form long, continuous tubes, the vessels.
Continuous tubes.
Yeah, the end walls between vessel members are often perforated or even completely dissolved, creating direct, open pipelines. Think of them as wider, more open pipes compared to tracheids, allowing for faster bulk flow of water.
So angiosperms have these superhighways for water. What else is in xylem? You mentioned fibers and parenchyma.
Right. Xylem also contains xylem fibers, which are basically sclerenchyma fibers mixed in. Their main role is additional structural support reinforcing the plumbing.
Okay, more strength.
And xylem parenchyma. These are the living cells within the xylem. They have thinner walls, store food reserves like starch, and are crucial for the lateral or sideways movement of water and minerals, especially in structures called rays.
So storage units and side connectors.
Pretty much. And just a quick note on development, the first xylem to form is called protoxylem, usually with narrower elements, and the later formed xylem is ataxylem with wider elements. Their arrangement differs too in darch and stems, meaning protoxylem is towards the center.
And exarchan roots?
Exarchan roots, exactly. Protoxylem towards the outside reflects how those organs grow.
Fascinating detail. Okay, that's water transport covered. Let's switch to phloem. This is about moving sugars, right?
Mm-hmm.
The food produced during photosynthesis.
Exactly. Phloem is the plant's food delivery network. It transports sugars, primarily sucrose, made in the leaves, the source, to other parts of the plant where they're needed for energy or storage, the sinks, places like roots, fruits, growing tips.
So restocking the pantry and fueling growth.
You got it. And like xylem, phloem in angiosperms is a complex tissue with multiple cell types: sieve tube elements, companion cells, phloem parenchyma, and phloem fibers.
Sieve tube elements and companion cells you mentioned, they work very closely together.
Incredibly closely. They're functionally inseparable. Sieve tube elements are the actual conducting cells. They're long cells joined end to end to form sieve tubes.
Sieve tubes. Why sieve?
Because their end walls are perforated by numerous pores, forming structures called sieve plates. These allow the sugary sap to flow relatively easily from one cell to the
Oh,
next.
like a sieve. Okay.
But here's the really weird thing. Mature sieve tube elements lack a nucleus, ribosomes, and a large central vacuole. They're basically just tubes filled with cytoplasm connecting through those sieve plates.
How do they stay alive and function then?
That's where the companion cells come in. Each sieve tube element is intimately associated with one or more companion cells, connected by numerous plasmodesmata tiny channels linking their cytoplasm.
So they're linked.
Very much so. The companion cell is a specialized parenchyma cell that does have a nucleus and all the necessary metabolic machinery. It performs all the life support functions for itself and its associated sieve tube element. It's thought to control the loading and unloading of sugars and maintain the pressure gradient needed for transport.
So the sieve tube element is the delivery truck, and the companion cell is the driver, navigator, and mechanic, all rolled into one.
That's a fantastic analogy. Exactly. The sieve tube is the conduit, but the companion cell runs the show.
Makes sense.
Hmm.
What about the other parts, phloem, parenchyma, and phloem fibers?
Phloem parenchyma cells are generally elongated parenchyma cells mixed in. They primarily function in storing food reserves, starch, fats, tannins, resins, and also help with lateral transport. Interestingly, they're often absent in monocots.
Storage depots along the route.
Yep. And phloem fibers, also called bast fibers, are sclerenchyma cells that provide structural support and protection to the delicate sieve tubes and companion cells. They're often abundant in secondary phloem.
And some of these are useful to us, right? Like jute or flax?
Exactly. Jute, flax, hemp, those are all commercially important phloem fibers, valued for their strength.
Yep.
And just like xylem, phloem also has early forming protophlloem and later forming metaphloem.
Okay, we've got the individual tissues down, simple and complex. Now, how are these actually arranged inside the plant? It's not just random, right? There's a system.
Absolutely not random. The way these tissues are organized into systems is crucial for how the whole plant works. Based on their location and function, we group tissues into three main tissue systems.
Three systems, okay.
There's the epidermal tissue system, which is the outer protective covering. Then the ground tissue system, which makes up the bulk of the plant body and handles things like photosynthesis and storage. And finally, the vascular tissue system, containing the xylem and phloem for transport.
So skin, filling, and plumbing wiring. Back to the building analogy.
Exactly. Outer walls, epidermal, internal structure and rooms, ground, and the utilities, vascular, all organized to work together.
Let's start with the outside. The epidermal tissue system, the plant's first line of defense.
Precisely. It forms the outermost layer of the primary plant body, young roots, stems, leaves. Its main job is protection against water loss, physical damage, pathogens.
And what's it made of?
It's mainly composed of the epidermis itself, which is usually a single layer of tightly packed cells, but it also includes specialized structures like stomata, the pores for gas exchange, and various epidermal appendages like hairs, called trichomes on shoots, and root hairs on roots.
So the epidermis is the main skin. What are those cells like?
They're typically flattened, somewhat elongated parenchyma cells, fitting together tightly like puzzle pieces with few gaps. They usually lack chloroplasts, except for the guard cells of stomata.
And they produce that waxy cuticle ray.
Yes, the epidermal cells of stems and leaves secrete that waxy cuticle layer on their outer surface.
Like a built-in raincoat to stop the plant drying out, except on roots, of course.
Exactly. The cuticle is crucial for preventing water loss from the aerial parts. It's generally absent on roots because, well, roots need to absorb water. Think of it like wax polish on a surface provides a barrier.
Okay. Then you mentioned stomata, those tiny pores, mostly on leaves, that control gas exchange.
Vital structures, yes. Stomata regulate the exchange of gases, CO2 coming in for photosynthesis, O2 going out, and also transpiration, the release of water vapor.
And they can open and close.
They can. Each stoma, which is just the pore itself, is flanked by two specialized guard cells. These guard cells control the size of the pore opening.
How do they do that?
It's based on water pressure, or turgor. When the guard cells fill with water, they bow outwards, opening the pore. When they lose water, they become slack and the pore closes. They have differentially thickened walls, thicker on the side facing the pore, thinner on the outside, which helps this mechanism.
Clever design. And they're usually bean-shaped.
Bean-shaped in most dicots, yes. In grasses and some other monocots, they're more dumbbell-shaped. And interestingly, guard cells do have chloroplasts. Sometimes surrounding epidermal cells are also modified as subsidiary cells, which help the guard cells function. The whole unit, pore, guard cells, subsidiary cells is called the stomatal apparatus.
Little controllable mouths all over the leaf surface. Amazing. What about the other epidermal things, the hairs?
Right, the appendages. On roots, you have root hairs. These are tiny unicellular extensions of the epidermal cells right near the root tip.
And they massively increase the surface area.
Hugely. They dramatically increase the root's ability to absorb water and dissolve nutrients from the soil, like millions of tiny drinking straws.
And on stems and leaves, you get trichomes.
Yes, trichomes are the hairs on the shoot system. They can be unicellular or multicellular, branched or unbranched, glandular, secreting stuff, or non-glandular. Hugely diverse.
And what do they do?
Lots of things. Some reduce water loss by trapping a layer of moist air near the surface. Some deter insects or grazing animals. They might be sharp or sticky or release irritating chemicals. Some reflect excess sunlight. Very versatile.
Okay, so the epidermal system is the protective, interactive outer layer. What about the ground tissue system, the filling?
The ground tissue system includes basically everything that isn't epidermal or vascular. It forms the bulk of the primary plant body.
And it's mostly parenchyma.
Primarily parenchyma, yes, but it can also include collenchyma and sclerenchyma for support in specific areas.
Where do we find it?
In stems and roots, it makes up regions like the cortex between the epidermis and vascular tissue, and the pith, the central core, if present. Also the pericycle, just inside the endodermis. In leaves, the ground tissue is specialized for photosynthesis and is called the mesophyll.
Ah, the mesophyll. That's where the chloroplast action is.
Exactly. The mesophyll in leaves is packed with thin-walled parenchyma cells loaded with chloroplasts. It's the primary solar panel and sugar factory of the plant. So ground tissue is doing storage, support, and crucially in leaves, photosynthesis.
The internal workhorse. Okay, that leaves the vascular tissue system, the transport network.
Right. The vascular system, made of xylem and phloem, runs throughout the plant, connecting roots, stems, and leaves. It's the internal plumbing and delivery service.
And xylem and phloem are usually found together in strands.
Yes, they typically occur together in discrete units called vascular bundles, like bundled pipes and wires running through our building.
And these bundles can be open or closed. What does that mean again?
It relates to secondary growth potential. In dicot stems, the vascular bundles usually have a layer of vascular cambium sandwiched between the xylem and phloem. This cambium is meristematic and can produce more xylem and phloem, allowing the stem to thicken. These are called open bundles, open to further growth.
Like expandable pipe bundles.
Exactly. In monocots, the bundles lack this cambium. They're formed during primary growth, and that's it, no secondary thickening from the bundles themselves. These are called closed bundles.
Like prefabricated, non-expandable bundles.
Good way to put it.
And the arrangement within the plant, radial and conjoined.
Right. Those describe how xylem and phloem are positioned relative to each other. In roots, the typical arrangement is radial. Xylem and phloem lie in alternating strands on different radii, often forming a central core, like spokes of a wheel, maybe?
Okay, alternating spokes in the root.
In stems and leaves, the arrangement is usually conjoint. Xylem and phloem are found together side by side within the same bundle.
Side-by-side partners.
And in these conjoint bundles, the phloem is typically located towards the outside of the stem or the lower side of the leaf, and the xylem is towards the inside or upper side.
Okay, radial in roots, conjoint in stems and leaves. This framework of tissue systems really helps visualize how it's all put together. Now, can we dive into the specific differences between dicots and monocots in different organs, seeing how these systems are actually laid out?
Absolutely. Looking at cross-sections is the best way. Let's start with a typical dicotyledonous root, like a sunflower root.
Okay, dicot root. What do we see from outside in?
Outermost layer is the epiblema, or epidermis, bearing those unicellular root hairs for absorption. Then you move into the cortex, which is usually a broad zone of parenchyma cells, often with intercellular spaces, mainly for storage.
The storage zone.
Right. The innermost layer of the cortex is very distinct. It's the endodermis, a single layer of tightly packed, barrel-shaped cells.
And this has those Casparian strips, the waterproof seals.
Exactly. The Casparian strips are bands of waxy suberin within the radial and tangential walls of the endodermis cells. They block water from moving between the cells, forcing it to pass through the cell membrane. This gives the plant control over what minerals enter the vascular cylinder.
Like a selective filter or checkpoint.
Precisely. Inside the endodermis is the pericycle, often a few layers of cells, usually parenchyma. This layer is important because it's where lateral roots originate, and in dicots, it contributes to forming the vascular cambium later for secondary growth.
So the source of branching and thickening.
Yes. Then you get to the vascular cylinder itself. In dicots, the very central region is usually small or even absent. The xylem typically forms a star-shaped pattern in the center, with maybe 2 to 4 arms.
A central star of xylem.
And the phloem patches are located between the arms of the xylem star. This is that radial arrangement. The tissue between the xylem and phloem is called conjunctive tissue, usually parenchyma. And later, that vascular cambium develops between the xylem and phloem.
Okay, so outer skin with hairs, storage cortex, waterproof endodermis, pericycle for branching thickening, then a central star of xylem with phloem tucked in between. Got it. How does a monocot root compare?
Monocot roots share the basic layers:
epiblema, cortex, endodermis with Casparian strips, pericycle. But the vascular cylinder arrangement is quite different.
Different how?
The most striking difference is usually the pith. Monocot roots typically have a large, well-developed pith made of parenchyma right in the center.
So a big central core, not a small one like the dicot.
Exactly. And surrounding this large pith, you find numerous xylem and phloem bundles arranged in a ring. There are usually many more xylem bundles than in dicots, often more than 6, a condition we call polyarch.
Polyarch, meaning many arches or bundles.
Right, so you have a ring of alternating xylem and phloem bundles surrounding a large central pith. And crucially, monocot roots don't develop a vascular cambium, so they don't undergo secondary growth.
Okay, key differences. Many xylem bundles, polyarch, in a ring around a large central pith in monocots versus fewer xylem bundles, usually 2-4, in a central star with little or no pith in dicots, and no secondary growth in monocots.
You've nailed the key distinctions for roots.
Great, let's move up to the stem. A young dicot stem, what's the layout there? Okay, young dicot stem. Outermost is the epidermis, typically with a cuticle, maybe some trichomes, maybe a few stomata. Beneath that is the cortex.
Cortex again, is it layered?
Often, yes. Just under the epidermis, you frequently find a few layers of collenchyma, the hypodermis, providing flexible support.
The supportive layer for the growing stem.
Exactly.
Yeah.
Deeper in the cortex, you usually have parenchyma cells, often with intercellular spaces, for storage and photosynthesis if it's a green stem. The innermost layer of the cortex might be distinguishable as an endodermis, sometimes called a starch sheath, because the cells accumulate starch grains.
Starch sheath, okay. Inside that.
Inside the endodermis starch sheath, you have the vascular cylinder. Just inside, associated with the vascular bundles, is the pericycle, which in stems might be present as patches of sclerenchyma, often above the phloem, providing more rigid support.
Sclerenchyma caps on the bundles.
Sort of, yeah, often described as semi-lunar patches. The main feature, though, is the arrangement of the vascular bundles themselves. In dicots, they're arranged in a distinct ring.
A ring of bundles, okay.
And each bundle is conjoint, xylem and phloem together. And importantly, open, meaning there's vascular cambium between the xylem and phloem, allowing for secondary growth later.
Open for growth. And position.
Typically, phloem is towards the outside, xylem towards the inside. And the xylem development is endarch. The protoxylem, first formed, is towards the center. The metaxylem, later formed, is towards the outside.
Got it. What's in the very middle?
In the center is the pith, usually quite large in a young dicot stem, composed of parenchyma cells, often with large intercellular spaces. And connecting the pith to the cortex, running between the vascular bundles, are strips of parenchyma called medullary rays or pith rays. They help with radial transport.
Okay, summarizing the dicot stem. Epidermis, layered cortex, collenchyma, parenchyma, maybe starch sheath. Then a ring of open conjoint vascular bundles, flown outside and dark xylem inside, all surrounding a large central pith connected by medullary rays.
Phew.
That's a perfect summary.
Now, how does the monocot stem contrast with that organized ring structure? I remember you said it was more scattered. Very different indeed. Monocot stem, you have the epidermis on the outside. Just beneath it, there's usually a hypodermis, but here it's typically made of sclerenchyma, providing rigidity right away.
Sclerenchyma for support, not collenchyma.
And then you get into the ground tissue. And the key difference is the vascular bundles are scattered throughout this ground tissue. There's no distinct ring arrangement and no differentiation into cortex and pith. It's just a mass of ground tissue, usually parenchyma, with bundles embedded in it.
Scattered like chocolate chips in a cookie.
Haha, yeah, that's not a bad visual. The bundles towards the periphery are often smaller and more numerous than the larger ones towards the center.
And the bundles themselves, are they open or closed?
They are conjoint, like in dicots, but they are closed. There's no vascular cambium, so no secondary growth and thickness from these bundles in monocots.
Closed bundles, scattered arrangement. Any of their features?
Each vascular bundle is usually surrounded by a prominent sclerenchymatous bundle sheath, giving it extra support. And monocot bundles often lack phloem parenchyma. Sometimes you also see large water-containing cavities within the bundles formed by the breakdown of some protoxylem.
Okay, so monocot stem, sclerenchyma hypodermis, ground tissue without distinct cortex pith, numerous scattered closed vascular bundles, each with a sclerenchyma sheath. Very different from the dicot's ring.
Fundamentally different organization, yes.
Right, last stop. The leaf. Let's do the typical dicot leaf first, the kind that's usually held horizontally. Dorsiventral, is that the term?
Dorsiventral, yes, meaning it has distinct upper dorsal or adaxial, and lower ventral or abaxial surfaces that are structurally different.
Okay, Dicot leaf structure.
You have an upper epidermis and a lower epidermis, both usually a single layer of cells covered by a cuticle. Stomata, those pores, are typically much more numerous on the lower epidermis.
Fewer on top, more underneath. Why?
Reduces water loss. The lower surface is usually shaded, cooler, and less exposed to direct sun and wind.
Makes sense. What's between the epidermises?
That's the mesophyll, the ground tissue of the leaf, packed with parenchyma cells containing chloroplasts. This is the primary site of photosynthesis.
And it's layered in dicots, right? Palisade and spongy?
Exactly. Typically, just below the upper epidermis, you have the palisade parenchyma, or palisade mesophyll. These are elongated, column-like cells arranged tightly in one or more layers, packed with chloroplasts. They're oriented perfectly to capture sunlight hitting the upper surface.
Like tightly packed solar panels.
Good analogy. Below the palisade layer is the spongy parenchyma, or spongy mesophyll. These cells are irregularly shaped, often rounded or oval, and they're arranged loosely with lots of large intercellular air spaces connecting to the stomata.
Spongy. For gas exchange.
Precisely. These air spaces allow CO2 to diffuse easily from the stomata to the photosynthesizing cells, and O2 to diffuse out.
So, palisade for light capture, spongy for gas exchange. What about the veins?
The vascular bundles, or veins, run through the mesophyll. They contain both xylem and phloem for transport of water to the cells and sugars away from them. The size of the bundles varies depending on the size of the vein, forming that characteristic net-like or reticulate venation pattern in dicots.
Network of veins.
Each vein is usually surrounded by a layer of compactly arranged cells called the bundle sheath. Within the vein, xylem is typically located towards the upper adaxial side and phloem towards the lower abaxial side.
Okay, dicot leaf, upper and lower skin with cuticle, more pores underneath, layered filling, palisade on top, spongy below, and a network of veins embedded within, like a specialized sandwich.
Perfect description.
Now, how does the monocot leaf, often more upright, compare? Isobilateral.
Isobilateral, yes, meaning both surfaces are often structurally similar because they receive similar amounts of light.
So stomata are probably on both sides.
Often, yes. Stomata are typically present on both the upper and lower epidermis, sometimes in roughly equal numbers.
And the mesophyll, is it layered like the dicots?
Usually not. In most monocot leaves, the mesophyll ground tissue isn't differentiated into distinct palisade and spongy layers. It's often just composed of more or less uniform parenchyma cells containing chloroplasts, filling the space between the two epidermises.
Simpler filling. Any other special features in monocot leaves? I remember something about grasses. Bulliform cells.
Yes, bulliform cells, very characteristic of grasses and some other monocots. These are groups of large, thin-walled, empty-looking cells, usually found in the upper epidermis, often in rows alongside the veins.
And they help the leaf roll up.
Exactly. They're involved in the rolling and unrolling of the leaf in response to water availability. When water is plentiful, the bulliform cells are turgid, full of water, and the leaf blade is flat and open. When water is scarce, they lose turgor, become flaccid, and this causes the leaf to curl or fold inwards, reducing the exposed surface area and conserving water.
Like tiny hydraulic hinges.
That's a brilliant way to think of them.
And the veins in monocots.
The vascular bundles run parallel to each other, reflecting the parallel venation typical of monocots. They're usually fairly similar in size, unlike the varying sizes in a dicot's net venation, and they are also enclosed by a bundle sheath.
Okay, monocot leaf. Often similar top and bottom surfaces with pores on both, uniform mesophyll filling, parallel veins, and in grasses, those special bulliform cells for rolling up.
You've captured the essence of it perfectly.
Wow. We've really gone deep into the internal structure today, from the basic idea of cells making tissues to the different types of tissues, meristematic for growth, permanent for function, simple, complex.
And then seeing how those tissues are organized into the epidermal, ground, and vascular systems.
Yeah, and finally comparing the actual anatomical layouts in dicot versus monocot roots, stems, and leaves. It's incredibly intricate when you look closely.
It truly is. And just to quickly recap the big takeaways. Remember those levels of organization: cells, tissues, organs, tissue systems. The key split between dividing meristems and specialized permanent tissues.
And simple versus complex tissues, like parenchyma versus xylem or phloem.
Exactly. And then those contrasting features, the root structure, the stem bundle arrangement, ring versus scattered, open versus closed, the leaf mesophyll layering. These are fundamental differences between dicots and monocots.
It really hits you how these internal blueprints directly lead to what we see on the outside, doesn't it? That broad dicot leaf with its palisade layer catching sun, the sturdy, thickening trunk of an oak tree because of its cambium, the way grasses spring back after mowing.
It gives you a whole new appreciation for the efficiency and, frankly, the elegance of plant design. Understanding that hidden architecture makes you see them differently.
Absolutely.
And it leads to a really interesting thought, something for you to maybe ponder later. How might these specific anatomical differences we've talked about, the scattered bundles in monocots versus the ring and dicots, different root systems, the leaf structures, how might these actually explain why these two groups are successful in different environments or why we use them differently?
Connecting anatomy to ecology and agriculture.
Exactly. Think about how a fibrous monocot root system compares to a dicot taproot for water access, or how the presence or absence of secondary growth influences how we use the stems for timber versus, say, grain. How does that internal design translate into real-world function and utility?
That's a fantastic question to chew on. It makes you realize this isn't just abstract biology, it has real consequences.
It really does.
So the next time you're looking at a plant, maybe take a second to picture that amazing internal world, the transport systems, the growth zones, the specialized cells, all working together. If this deep dive has sparked your interest, definitely explore more plant biology. There's so much more to discover.